Hello and welcome everyone to this podcast for epigenetics. In this podcast we're going to focus on the reversibility of the epigenome. If you remember last time I showed a picture here of my son and I said all of his cells are for the most part differentiated except for his stem cells. And I said that, remember I said this mantra, differentiation is unidirectional. And so that his cells could not go back to being an embryonic cell because his pluripotent genes, those that maintain the pluripotent state, are off. If you were to take, say, an epithelial cell from him, there's going to be some genes on and off to perform the specific functions of only the epithelial cell. You'll also have another subset of genes that are poised. They're ready to be turned on if necessary. These might be genes needed for DNA repair. They might be genes needed for remediating various toxins that might enter the cell. So they're poised. However, all of the cells, the epithelial cells, all the cells still retain genetic information to make an embryo. It's good for him and it's good for all of us that our cells cannot go backwards. Now what we're going to talk about though is how sometimes we can make these cells go backwards. We can force the genomes in any one of his cells or my cells or your cells to sort of remember their past and could set the stage, the genetic stage, for embryonic development. So how can we do that? Your book mentions 4, and I'm going to throw in a 5th as well. So 5 ways to reverse epigenome. The first is somatic cell nuclear transfer. And that's what we're going to talk about in this podcast. And then, depending on how much time we have, we'll talk about some of these others. We can reprogram it by cell fusion. You can reprogram it by providing certain cellular extracts. And the last one, which we'll certainly get to, are these IPS cells, which stands for induced pluripotent stem cells. These first 4 that I'm showing here are ways that we can reverse the epigenome experimentally. But unfortunately, the epigenome can also be reversed in some forms of cancer, where a fully differentiated cell can reverse and become an undifferentiated cell and can lead to cancer. Now this we won't talk about till towards the end of the semester, so you'll have to keep coming back to hear that story. But let's move on to talking about somatic cell nuclear transfer. Alright, so we're going to talk about somatic cell nuclear transfer, and we will abbreviate that throughout this podcast as SCNT. Now about 60 years ago, a paper was published by Briggs and King, and they worked with frogs. A lot of this early work was done with frogs. And what they did was they took an oocyte, a frog oocyte, and they removed the nucleus. They then took a frog embryo, a fairly young embryo, and took a nucleus from it. So it would have been a diploid nucleus because it's an embryo. And they would take that frog embryo nucleus and they placed it in here. And what they discovered when they did this was that they would get some of the frogs that would form tadpoles. So somehow the genetic information in this frog embryo, when placed in this oocyte, was enough to remind this oocyte and the DNA in this diploid nucleus from the embryo, remind it how to become a tadpole. It wasn't incredibly efficient. Not everyone formed a tadpole, just some of them did. But it was enough to prove the point that the cell could somehow, or the nucleus could somehow remember. They also, not they, but somebody else, in the 1960s, took the same denucleated oocyte, well not the same one of course, but they took a frog oocyte, removed the nucleus, and then they took a cell from a frog's intestine and removed that nucleus and placed that into the nucleus, into the oocyte. And again, what they found was that some tadpoles would form. Again, it wasn't incredibly efficient, but they did get some that form, again proving that even a fully differentiated cell, the nucleus could remember. Because you might argue, well, the embryo is much closer to that undifferentiated state, and so maybe it was easier for it to quote-unquote remember. But these were fully differentiated cells, indicating that all the genetic material that was necessary is never lost. It stays with us forever. It's like a bad tattoo, or a good tattoo. Now, early mammalian attempts all failed. They tried it with a variety of different kinds of organisms, and it always failed. They didn't know why it would work for frogs, but not, say, mice or pigs. But they reasoned that the differentiation process in mammal cells was much more complicated, and by the time you made it to a fully differentiated cell, that it had gone too far to remember what it was like, so to speak, to be that early zygote. We now understand that the reason these early experiments failed was largely due to epigenetics. The reprogramming of the epigenome is seen in mammals and not in frogs. And so this helped explain some of the reasons why it failed. Now, of course, it didn't always fail, because we wouldn't be talking about it right now in this epigenetics class. So it must have somehow worked, and it did. And back in 1996, this was my first year as a graduate student, so I remember it well, there were some scientists. Okay, and they worked with two different kinds of sheep. The first was a Scottish blackface sheep. I was going to marvel you with my mad drawing skills here and draw a picture of a sheep, but I thought I would spare you that and just draw this circle. So just pretend this is a sheep. It's probably actually a little bit better than what my drawing would have looked like. They also worked with another sheep called a Finn Dorset. And what they did with the Scottish blackface sheep is they isolated an oocyte. It being a oocyte, it was only a haploid genome. But they removed it and discarded it. And now we had an oocyte that was enucleated. It still had all the cytoplasmic factors in it that were found. The only thing removed was the nucleus. With the Finn Dorset sheep, what they did was they removed an epithelial cell from the udder. Now this would have been a diploid nucleus because it's a fully differentiated somatic cell. And they removed this nucleus and discarded the enucleated cell here, the epithelial cell. They then allowed the nucleus from the Finn Dorset sheep to fuse with the enucleated oocyte from the Scottish blackface sheep. And their hopes were that this diploid nucleus would resemble a fertilized egg, having a haploid nucleus from the maternal source and a haploid nucleus from the paternal source. They then took this and placed it into a surrogate mother. And this surrogate mother was another, not the same one, but another Scottish blackface sheep. And they allowed it to develop. And what resulted was Dolly, a Finn Dorset sheep. Because all the genetic material found in this epithelial cell, when placed into the oocyte, was somehow reprogrammed and remembered, so to speak, its embryonic history. And even though it was placed inside of the Scottish blackface sheep, the progeny that formed was, of course, of the Finn Dorset sheep. And they named it Dolly. Now, even though nuclear speaking, the nuclear DNA from this sheep here, there are other factors that certainly the Scottish blackface sheep here provided from the cytoplasm of the enucleated oocyte. And we'll talk a little bit about those. Now, since Dolly was made in this way using somatic cell nuclear transfer, and again, somatic cell because we take it from a somatic cell and we transfer it, nuclear transfer, we transfer it to a oocyte. This has been reconfirmed in sheep, cows, rabbits, horses, I think I read that somewhere, and mice and pigs. And they've even been able to do some interspecies. Now the interspecies, for ethical reasons, they haven't allowed it to go all the way to this form here, but they have been able to take very species, closely related species, and mix the somatic nucleus into the other species oocyte, and you do go to the blastocyst stage before they stop it. So now what I want to do is I want to do some comparisons of this SCNT formed embryo and an embryo formed through natural fertilization. So up here I'm going to put natural fertilization, and all I mean by that is using an oocyte and a sperm. And so here's our oocyte. It has a 1N nucleus, a haploid nucleus. It also has this protein factor called MPF. And MPF stands for maturation promotion factor. MPF does a few things. It's responsible for the meiotic pause, more specifically the meiotic 2 pause during metaphase. Remember that as the egg develops, it's paused during meiosis 1 until it's released during ovulation. Once it's released during ovulation, meiosis starts again, but then it pauses again at meiosis 2. And it doesn't complete meiosis until fertilization occurs. So MPF helps keep this in this meiotic 2 metaphase pause. MPF also prevents nuclear laminin from forming. So it keeps that nuclear membrane from being a mature membrane. And because the mature nuclear membrane doesn't form, the genetic material is just held together by small vesicles. So upon fertilization, when the sperm enters the egg, and remember we talked about how once that happens, we will form these 2 pronuclei, one from mom and one from dad. At this point here, MPF, its expression is downregulated. We don't see as much MPF. Meiotic pause is released, so it re-enters meiosis. The egg nucleus does. The oocyte genome does. But this doesn't just happen because the sperm entered the egg. When the sperm enters the egg, what happens is a calcium wave moves across the cytoplasm. It's a pretty quick wave, and if you've taken a cell biology class, or if you've taken a developmental biology class, which I would highly recommend, this calcium is a very important second messenger for many different cell signaling pathways. And so as this calcium wave occurs, it decreases MPF, which allows the meiotic pause to be released, and now the lamins form. I'm going to erase part of this so that we can see what happens with somatic cell nuclear transfer. So SCNT. You have the donated oocyte, and it being an oocyte, you're still going to have the MPF in there. It's enucleated. Remember, it doesn't have the nucleus. But then you add that diploid nucleus from a somatic cell to it. So what you have is this diploid nucleus in here with MPF. As shown here, MPF is still high. The meiotic pause is still in place. The lamins don't form. You don't create the pronuclei. And these things don't occur because calcium isn't released from the stores within the cytoplasm of the oocyte. So you don't have this calcium wave moving across the fertilized egg, or in this case the SCNT egg. But what they can do is they can use something called a calcium ionophore. And what this will do is it will open calcium channels and allow for that calcium to be released. When that happens, interestingly, you will form these pronuclei. But what's particularly interesting is you may form more than 2. There's something about the program that once this calcium wave comes through, that these pronuclei form. Now, not all have DNA in them. And there's no way to separate, at this point, maternal versus paternal DNA components. So I suspect, though I don't know if they know for sure, but I would suspect all the DNA is in one of these pronuclei. There's no reason to think that they would be separated. Also, what happens once you have this artificial calcium wave come through, the lamins form, MPF is downregulated. The meiotic pause isn't necessarily released because we're not dealing with anything that is in a meiotic pause. I want to make a couple notes down here just to remind ourselves. Remember that the sperm and egg up here through this process is mostly absent of epigenetic markers, with the exception of imprinted genes. Remember, as they are made, it reverses or demethylates the genome except for those imprinted genes. This diploid DNA from the epithelial cell or from any cell is heavily methylated. So in some ways you can think of this heavily methylated DNA in this nucleus here as being this foreign invader, that the oocyte isn't expecting this. You wouldn't necessarily think it would know what to do with it. But we do know that longer exposures to MPF by holding off on the calcium wave will help demethylate it. We also know that serial nuclear transfers help. And what I mean by that is if you add the diploid nucleus here and you let it sit for a certain amount of time, you remove this nucleus and you add it to a new oocyte, and you repeat that a few times. That continuous and prolonged exposure to the different cytoplasms of different oocytes somehow helps to demethylate the DNA found in this donor nucleus.